Hi everyone, welcome back to another virology lecture. So today we are going to be talking about antivirals. And before we get into antivirals, I wanted to share with you the updated Johns Hopkins Coronavirus Resource Center. So Johns Hopkins has been updating this map a lot throughout this pandemic, and they came out with a recent update that added a really cool new feature. So this front tab is the tab that we're used to, one that we've spent a lot of time looking at throughout the course of this semester and throughout the course of the pandemic. So as you can see, the total confirmed cases are nearing 2 million across the world, and we still have the representation of the cases as the red dots, and the larger the red dot, the more total confirmed cases. But one of the cool new things that has been added has been this US map. And in this US map, they have a really cool heat map feature that they added. So let's go ahead and zoom in here. We'll zoom in on the Indiana area. And Indianapolis is definitely a hotspot, so why don't we go ahead and click on one of the counties. So here is Marion, Indiana, and they added this really cool new feature where not only does it tell you the confirmed cases and deaths, but there's this infographic that you can click on as well. And the infographic takes you to a new tab and it gives you even more data. And it's really interesting because it breaks it down based on the health facts and the infrastructure and based on population. And you have your policies and cases of HRSA. It has the calculated fatality rate for that county. So really cool new features that they've added. And then I believe that this tab is also new, this critical trends tab. So it has a lot of different questions that people might be interested in. So where are cases increasing? How is the outbreak growing? And then how does mortality differ? And then of course we spend a lot of time talking about flattening the curve. And so this question of has the curve flattened or not? So they do a really nice job of having a 5-day moving average of everything. And so this dark green line, the one that is all the way on the top, that is us. So that is the US. So you can see that we're starting to finally take a decrease. So finally starting to flatten that curve, and hopefully this decrease continues. So speaking of pandemics and speaking of viruses of HRSA, we can't have a virology class without talking about antivirals. So what we're going to do is we are going to start with a general introduction, and then we're going to move into drug development. So we're going to talk about some history, some challenges, the different screens that we have, and then in day 2, what we will do is we will actually get into parts 3 and 4. So we'll get into examples of antiviral drugs, we'll talk about their specific mechanisms of action. We'll get into a little bit about how does resistance occur for the examples that we'll talk about. And then I've added a new section to this lecture so that we can spend some time talking about the different therapeutics that are being used for the current pandemic, so for SARS coronavirus 2. So let's go ahead and get started with a very general point. So when it comes to viruses, we really have 2 arms of defense, and that is our vaccines and our antivirals. And for vaccines and for antivirals, they definitely work in very different ways, and we have a topic devoted to each of these arms of defense. So after we wrap up with antivirals, we'll actually then talk about vaccines. So for vaccines, their main purpose is really to prevent viral disease, but they offer no therapeutic effect. And there is, of course, an exception to that because, as we know, there's an exception to everything, and that exception, of course, is rabies. So if you get bit by a rabid animal, you then get the rabies vaccine, and because rabies takes such a long time to get to the CNS, you have some time to get your vaccine and to build up immunity that way. So you can get therapeutic effects with rabies, but other than that, the whole purpose of vaccines is to prevent viral disease. And then, of course, our second arm of defense is our antivirals. And for antivirals, what they do is they stop infection once it has begun. So we have known about viruses for quite a long time, as we talked about in our first topic way back when the semester started in January, but it wasn't really until the 1950s that we started studying antivirals. And so currently, even though it is 2020, we currently only have about 100 antivirals that are at least available on the US market. So there are, of course, other ones. Some of them are not approved by the FDA, and there are others that are used in other countries but are not used in this country. But we have about 100 or so that are available on the US market. And most of our antivirals are against persistent infections. So if we take a look at the chart that we have here, on the x-axis here we have time, and then on the y-axis we have the number of approved drugs. And for these antivirals, they're actually split into the virus that they are used against. And so for our persistent infections, the majority of the drugs are against HIV. So HIV is this red line here. So we've got about 40 drugs against HIV, and then other persistent infections are hepatitis C virus, herpes viruses. There are, of course, exceptions on this list. So if you take a look at some of the viruses that are acute infections, that is going to include flu or influenza and RSV. So RSV is respiratory syncytial virus, and RSV is one of the most dangerous viruses for children, and in terms of viral diseases, it's definitely the most common that is found in children that can lead to death. So we, of course, have antivirals against those, but the majority of our antivirals are against persistent infections, and this goes back to the idea that most acute infections of HERS come on, and then you get a lot of viral production really fast, and then they come on fast and they leave pretty quickly as well. So our first antiviral was in 1963 that was first discovered, and that antiviral was an anti-herpes drug, and you are going to see me struggle this entire topic with pronouncing drug names. And the drug that was discovered in 1963 was idoxuridine. And for idoxuridine, it is a nucleotide analog, and you can probably guess from the name uridine that it is a deoxyuridine, so it's a uridine analog, and it is an anti-herpes drug. And it actually has an iodine that is added to the uridine, and what it does is it stops base pairing. So this was our first antiviral. And on this page, I actually have this review linked, and this figure is taken from this review. It's a fairly long review article, and it was published in 2016, but it does a really nice job of walking through all of the different antiviral drugs that we have. So you can probably tell based on this figure and based on the number of only about 100 antivirals that are approved in the US that we don't have a lot of antiviral drugs. So why are there so few of them? Why don't more exist? We certainly have a lot more antibiotics than that and a lot more other drugs. So why is it that there are so few antiviral drugs? So I'm going to give you a bunch of ideas that I have come up with. These are by no means all of the different ideas, so anything that I miss, as always, be sure to include in the comments below. So one thing that I'm going to write up here is genome instability. So many viruses are really good at evolving, if you will. We know that there are many RNA viruses. We know that RNA is less stable than DNA. And so there are a lot of mutations that can arise. So genome instability of HRSA means that you can have things like escape mutants and whatnot. In terms of viruses of HRSA, there's way more of them than anything else. So we have to deal with the high number of different viruses. Now, again, we're a little lucky. Not all of them infect us, but there's also issues of side effects. And many of these side effects of HRSA are due to the idea that every step in the viral life cycle engages host functions. So we have to find a way for these antivirals to be specific against the virus, but to not harm the host. Other issues that we have had in the past is this idea that some viruses can't be propagated. So if we can't grow them in the lab, it can of course be certainly more difficult to study them. And if it's more difficult to study them, it's going to be more difficult to develop or to find antiviral drugs against them. Other issues that we have are that for many viruses, there are no animal models. And the FDA actually requires that antiviral drugs that are approved and go on the market have to actually be tested and work well, and the safety and efficacy must be shown in at least 2 animal models. The only exception for that is smallpox. So we do have drugs against smallpox that are approved but have not gone through 2 sets of animal models, mostly because we don't have another set. But in general, we want to make sure that we have an animal model, and it's very difficult to find animal models for many of these viruses. And of course, other things, we have things like biosafety level 4s, right? Not everyone wants to work in the highest safety level as possible. And so that, of course, becomes a problem. The drug must be specific. And we all can also kind of think back to this idea of engaging host functions. If there are compounds that interfere with virus growth, they can also adversely affect the host. So lots of considerations when it comes to antiviral drugs. And again, I encourage you to think about other ideas that I have not listed here and then add them to our discussion below. So kind of moving forward, the next thing I want you all to think about is if you are designing antivirals, what would you target them against? Or what would their mechanism of action be? So there are a lot of correct answers to this, and I'm actually not going to answer this question. I'm going to show you a picture here on the next page, and then we will talk about some mechanisms of actions as we continue through this topic. But I am really curious to see what you all have to say and how you would design an antiviral. So I encourage you to pause the video and jot down your ideas below before you continue watching the lecture. So you probably have come up with a multitude of different ways that you could design an antiviral, and really pretty much any part of a virus's life cycle could be targeted. So I really like this image because what it does is it shows us the function. So essentially the step that we're at in the virus's life cycle. It shows us the picture of a virus's life cycle, and then it also talks about the example itself and then the virus. So as you can see in this image, we have different drugs that target attachment. We have drugs that target uncoding. We have drugs that target mRNA synthesis. We have drugs that can target protein synthesis and replication and assembly. So pretty much any part of the virus's life cycle that you can think of, we can probably target it. But again, just because we can target it doesn't mean that there are not inherent challenges that come with drug development. So let's now kind of transition into drug discovery. What do you all think are some of the challenges that are associated with drug discovery? And again, I encourage you to brainstorm and then jot down some of your answers and some of your ideas that you come up with in the comments below. So lots of different things that we can kind of bring into this discussion that we've already started talking about. So challenges of HRSA, animal models, side effects, right? Human models. Eventually you move into the phase of clinical trials. So you have to get humans to come in and to volunteer to do this. Cost is a huge thing. Time, efficacy, right? Popularity in terms of medical relevance. So a year ago, probably no one really was looking at antivirals against coronaviruses. And when I say no one, I'm of HRSA being very extreme here. There are groups that have studied coronaviruses and will probably continue to study coronaviruses forever. But the point is that it was not something that was pushed in the scientific community. So we definitely see that funding comes in waves, and that funding oftentimes goes to what is hot, what is sexy currently in the field. So that's definitely a challenge that comes with drug discovery as well. And in general, what kind of knowledge do we have about the virus? So I guess you can say, fortunately, for the current coronavirus outbreak, we have studied SARS, we've studied MERS, so we definitely didn't go into this completely blind. We at least were able to start somewhere and have some background knowledge, and we're able to go in with something. But I want to tell you a couple of key point challenges here with drug discovery, and they kind of deal with key points. So there's 2 key points that I'm going to tell you. And key point number 1 is that whatever the compound is, so whatever the drug is it must block viral production and viral replication completely. And so this idea that partial inhibition is not acceptable. And the reason of HRSA is that resistant mutants will arise. So this is, I say that it must completely block viral production and replication, but keep in mind, you can never get 100% blocking. Even when it comes to our antibiotics, you don't get 100% killing of the bacteria. You want enough where with the host immune system and everything else that is happening, the infection is able to be cleared. But again, our goal is still always to get 100% blocking. So part of the reason for that of HRSA deals with what's going to happen to your viral load over time. So let's draw a graph here. So we've got time, and then on the y-axis, that is very crooked, we have viral load. And so dosing is a very important part of drug development. So when the drug is given, so let's go ahead and add a dashed line here. So this is when the drug is administered. Ideally, of course, when that drug is administered, you want to see a viral load that goes down and then stays down. So this is the optimal, if you will, dose. Now, if you have an intermediate dose, you're going to see the viral load go down, and then you're going to see it increase. With a low dose, you're going to see maybe some decrease and then an increase of viral load as well. And so this is a very important key point and definitely a challenge when it comes to drug design, because the dose that you might need to decrease the viral load might be too high, and now it's no longer safe for human consumption. So there's definitely a balance. So that's the first key point. The second key point is this idea that many acute infections are of short duration. So if you have an infection that is of a really short duration, by the time you realize that you're sick, it's too late. And a really great example of that is when you have the flu. So oftentimes Tamiflu is given for the flu, and we'll actually talk about how Tamiflu works. But you have to take it within 24 to 48 hours. If you wait too long, then it's not going to help you. And so for many of these acute infections, you have to take it very early on. And so it goes back to this idea that the antiviral must be given at the right time. So then, this question another question that you could bring up is, why don't we give antivirals prophylactically, right? If we have this problem of you have a very short window to give an antiviral because an acute infection is so short, well, why not just give antivirals prophylactically to everyone? So, of course, it's kind of a bad idea, and the reason for that goes back to mutations. So, our arsenal of antivirals is already low. If we were to constantly be giving antivirals prophylactically, we would be definitely selecting for resistant viruses to these antivirals. And so, other issues that we have are this idea of no broad spectrum antivirals. Oftentimes we have lack of diagnostic tests. And one thing that I will say, if I back up a second, for this idea of giving antivirals prophylactically, we do not do that. And of course, there is an exception to that. And the exception to that is PrEP for HIV. So that is definitely a prophylactically given antiviral. So again, hopefully I've convinced you that there are a lot of challenges. And like I said, I look forward to seeing what else you all have to add in the comments below. So let's walk through a brief history here. So we started looking for antivirals in about the 1950s. And I know that I've already mentioned this, but you were probably wondering, well, why? Why was the 1950s so special that it took us until then to start looking for antivirals? So at the time, smallpox was still an issue, and that was definitely a push towards looking for antivirals. So at the time, what we knew that antibiotics work on bacteria. And so the question in the scientific community was, can we find things against viruses? And so in order to look for things against viruses, in the 1950s through the 1970s or so, we did something called blind screening. And blind screening is kind of exactly like what it sounds. What you end up doing is you test random things, and those random things could be chemicals, could be dirt, pretty much anything you have lying around, and you see if it stops growth. And so people were literally playing with dirt. It was not really necessarily a focus on discovering viruses or discovering viral mechanisms. You can imagine that this was a lot of work and very little success. The focus was just, can we find something that stops viral growth? So people would go on vacation, and scientists would have bags that they would bring with them, and they would scoop some dirt up. They would write up the coordinates of where they got the dirt from. When they got back to work, they would then test to see if there was anything that could work as an antiviral. So again, for these blind screenings, these were lots of work, but very little success. We did have success for a particular antiviral, and that is Amantadine. So Amantadine is shown here on the right. Amantadine is also sometimes known as Simitrel, and this was discovered in the 1960s by blind screening. And this was available for flu. It's not used very often just because there's a lot of resistance for it, but when they actually discovered it and then it actually started to be used, interestingly enough, we did not know the mechanism of action until the 1990s. So for, years we were using this drug without knowing how it worked. So I'm actually not going to tell you how it works yet, but later on in this topic we will come back to it, and then I promise I will let you know the mechanism of action of amantadine. So, all right, blind screening is dead. We don't really do that anymore, obviously, because lots of effort, not a lot of yield in anything. And so what do we do now today? There are a lot of different things that we now do instead. So now we know a lot more about viruses. So we look at the viral life cycles. And we definitely take more of a targeted approach. We can do a lot of recombinant DNA technology. So nowadays, we don't even have to be able to grow the virus in the lab as long as we know the sequence of the nucleic acid of the virus, so of its genetic information. We can use that and use recombinant DNA technology. So we can go ahead and do things like take viral genes, they can be cloned, and then we can analyze the gene products in atomic detail. And so oftentimes we will do this with what we think is the receptor. So glycoproteins are a really great example of this. And going back to this idea of looking at viral life cycles, we can of course target based on these. So rather than these blind screens, we are taking a much more specific approach. We of course have cell lines that we can work with. We can look at knocking out different parts of the cell line and getting a better understanding of how is the virus using the host cell, what is it doing in the host cell, and maybe kind of targeting approaches that way. So we talked about X-ray crystallography and cryo-EM earlier in the semester. Much of those techniques are used for the atomic detail and for some of the structural data that we do. So we definitely do things like computations as well, so a lot more computational work. All right, so kind of moving forward in terms of the path of discovery. As you will see, this takes a really long time, but we're going to talk about kind of the general path of discovery and what has to happen. So the first thing that we have to ask ourselves is there a medical need? And if there is, then we kind of move forward and we look to see what is the relevant mechanism, right? So there's a medical need. In this case right now, our medical need is the current coronavirus outbreak. What can we target? How can we target it? So we have this proof of principle is what we call it step. And then kind of moving forward from the relevant mechanism, then we move forward into screens. And we'll talk more about the screens that we have. They're very different than the blind screening that was done. And hopefully after some screens, we get what we refer to as some hits. And so these hits are going to be compounds that inhibit your virus. So from there, you can move into a potential drug candidate. And when we think about these hits and we think about them moving into a drug candidate, there's a lot of questions that we have to ask. So one of the things that is studied is bioavailability. So bioavailability deals with the idea of, will the compound get to the right place? We also have to think about the pharmacokinetics, which is my husband's favorite thing to think about as an almost pharmacist. And so, this idea of, will the drug or the compound persist long enough? So what is its half-life? How long will it be around? And then, of course, this idea of toxicity and specificity. So a lot of the safety parts. So will it be safe? And all of these things have to be taken into consideration, and all of that data kind of feeds into picking what our drug candidate is. And then eventually, of course, we get into testing. And the first type of testing that we usually do is in animal models. So this is a very, very long process. And I'm going to show you a figure that kind of takes some of these pieces, so this path of drug discovery, and does a nice job of showing us the time required. So on the x-axis here, we have the time required. On the y-axis, we have the representative number of compounds. So if we start here on the left, we've got a bunch of compounds that we've identified. We look for an antiviral effect in cells. So typically we start with cell lines. You can see a number of them are rejected. And then we say, okay, well, what's their toxicity in the cells? We see even more rejected. And then we can move into animals, and we'll find that more are rejected. We talk about low toxicity in animals, and then eventually, of course, we get to humans. So we have to look at toxicity in humans, antiviral effects, and then 10 to 15 years later, you can see this tiny little sliver of compounds that has been approved for general use. So this, as you can see, takes about 10 to 15 years to approve a drug. And that starts from 100,000 compounds that you may have made. If you are interested in the money, it takes about $100 to $200 million. So not only does it take about a decade, but it takes millions of dollars. And as you can see, most drugs fail. And it turns out that one of the steps that majority of drugs fail in is actually failing in humans. So if it does great in animal models and then we try it out in humans, most of the trials will actually end there. So thinking about drug trials kind of moving forward, this is a really cool image because it actually shows us the different phases or the paths from discovery to FDA approval to what we refer to as phase 4. So if we start on the top of the figure, and on the x-axis here we have years. So on the top of the figure we have our discovery. So after discovery and synthesis, we have a lot of preclinical that goes on. And this entire time from phase 1, which we'll talk about what phase 1 here is in a second, through phase 4, we have ongoing safety surveillance. And so any patient in any of the phases that is involved in any of those, their physician is constantly reporting data back because we constantly want to have a surveillance of safety, and this is when side effects and things like that are updated. So if we find a drug target that passes all the preclinicals, does well in animal models, and we are ready to move into humans, we go on to phase 1. And phase 1 is going to be about 30 people. And if the drug passes phase 1, so those 30 people or so, we then move on to phase 2. Phase 2 is often focused on efficacy and dosing. and it tends to be a larger number of people, so sometimes hundreds, and fully depends on the clinical trial. And then they move into phase 3, and phase 3 is like phase 2 but larger. And typically for phase 2 and 3, you'll have hundreds to thousands of people. Again, fully depends on the clinical trial. And by the time we get to the FDA approval or the FDA review, only about 1,000 people have tested the drug. And then once the drug is on market, so after the FDA approves it, we have phase 4. And phase 4 is what we refer to as post-market surveillance. And this is something that I did not actually learn about until last year. So as many of you know, I suffer from chronic migraines, and I tried a couple of different antibody drugs that had just come out on the market. And so they were technically a part of phase 4. So the FDA approved them. And I think within 2 months or so, I was able to get approval from my insurance to have them pay for that antibody drug, because they typically don't like to pay for new drugs. And so I unfortunately was not able to stay on the drug, even though it worked really well in terms of stopping my migraines. I had a lot of really bad side effects, and my arm swelled to like 3 times the size. So that was kind of an issue, but I'm actually a part of phase 4 for that drug, and all of my data my neurologist has reported back. So some of that data comes from me, and that is part of this post-market surveillance that occurs. So even though within 10 to 15 years, if you discover a hit and you get it to pass through everything, you are still looking at years of post-market surveillance. And sometimes we take drugs off the market because of what we find in post-market surveillance. So hopefully I am convincing you here that it is really hard and very difficult to get drugs approved. It's kind of amazing that it happens in general. So, all right, what kind of screening do we do then? So we said that blind screening is dead. So what are some of the ways that we identify potential drugs? And the key here is that regardless of how we do this, this needs to be high throughput. Right? It takes us long enough to get the dang thing approved anyways. We don't want to spend 10 years just looking for something and doing an experiment. We want it to be pretty high throughput. So there are a lot of different ways, and we're going to talk about 3 screens. We're going to talk about a mechanism-based screen, and then we're going to talk about a cell-based screen. And then last but not least, we're going to talk about an antiviral screen. So all of these methods are used, and what we're going to do is we're going to go through these one at a time, and I'm going to draw some pictures and then kind of walk you through how these mechanisms work. So the first one that we're going to start with is we're going to start with the mechanism-based screen. And the example that we're going to talk about for this mechanism-based screen is testing a viral protease. So many viruses have proteases that are involved at different parts of their life cycles. So we're going to model how can we use a viral protease. So our goal here is to target specifically a viral protease. And what we're going to do is we're going to develop a fluorescence-based assay. So what we're going to have is we're going to have some sort of a fluorescence tag, and this fluorescence tag is going to have a cleavage site that is attached to it. So this is going to be a short little peptide. So we've got a little protein, and that protein is going to have the cleavage site that the protease targets. And this is going to be attached to, usually we attach it to a bead. And so typically we have our viral protease, and this viral protease is going to recognize the cleavage site, and it's going to cut it, and when it cleaves it, that peptide gets cut in half, and that allows the fluorescent tag to essentially turn on. So what our goal then is to screen products. So we're going to draw a little... This is a really bad picture of a pill, but this is how I envision a pill, or actually instead of a pill, let's just call it a drug. It doesn't necessarily have to be a pill. And this drug is going to inhibit this protease. So it might do it by sitting in the active site. It might do it by sitting somewhere else in the protease. But this is going to instead lead to no cleavage and an intact tag, which is going to lead to no fluorescence. And so very quickly we can do a bunch of these, and what we're looking for is no fluorescence, and that tells us that that would potentially be a hit. So the second example that we're going to talk about is cell screening. So for cell screening, we can actually take advantage of bacteria and of tetracycline efflux proteins. So we're going to draw a membrane here. And this is going to be the inside of the cell. This is the outside of the cell. And we're going to draw a tetracycline efflux pump. And this actually makes bacteria resistant to tetracycline. So when we have this tetracycline efflux pump, because we are interested in looking for something that stops a viral protease, we know that viral proteases have specific cut sites. So what we're going to do is we're going to manipulate this tetracycline efflux pump, and you'll notice that when I drew it with these loops on the inside and the loops on the outside of the cell. So we are going to take one of those loops and change the amino acids there. And so this is now going to be our viral protease cleavage site. And if we cut that, that means that we will break the protein, which means the bacteria will not be resistant to tetracycline. And so that is how we actually screen our potential drug candidates. So what we're going to do is we're going to have some LB plus tetracycline plates, and we're going to do two scenarios. We're going to do a scenario where the drug does not work and the viral protease instead works. So here's our cleavage site. And then our second example will be our hit. So we'll do hit and miss. So if we have a viral protease, and let's say that we have drug number 1. If drug number 1 does not work against the viral protease, then that little loop will get chewed up. So that is now gone. And when that happens, that breaks the tetracycline efflux pump, which means that we will get no colonies. And the reason we get no colonies is because those bacteria have no way of pumping out the tetracycline, and so they end up dying. Now, on the bottom example here, let's say we have our viral protease, and we've got drug number 2. And now maybe this drug sits in the active site, or it somehow stops the protease. And so now that cleavage site does not get cut. And because that cleavage site does not get cut, we now have a fully functional tetracycline efflux pump, which means that we will get colonies that grow on our plate, telling us that this drug is a hit and a candidate. All right, so the very last example that we are going to talk about is antiviral screening. And for antiviral screening, this is going to be not only faster, but it's also going to be more high throughput than blind screening. And so what we can do is we can create chemical libraries. We can use natural products. We can do things like structure-based design. And oftentimes the structure-based design is going to be done with computational approaches as well. And you can screen up to 10,000 compounds a day. And it's pretty cool because when we do antiviral screening, we actually use these giant robots. So here is an image of a robot, and this robot is actually able to fill. So here's an example of a robot filling, nice scientific photo of a blue unknown solution here, but it does a nice job of at least showing us how it works. And what we do is not only do we use robots, but we also use micro titer plates. And micro titer plates come in different sizes, so you can have 96 well plates. So this right here, this clear one is a 96 well plate. This one right here in this image is also a 96 well plate. You can also have a 384 well plate, which is the largest well plate that I have ever used, and that is shown here in the middle. But you can also go up to a 1,586 well plate, which is the one shown on the top. It is very, very tiny. So I have worked with a robot before. It was not as large as the one shown here in this image. It takes quite a bit to actually program it, but it's really nice because you don't have to worry about being aseptic because it does it for you. So it's pretty amazing. All right, so now that we've covered screening, let's get a little into part 3. So for part 3, we're going to talk about examples of antiviral drugs. And we're going to save most of the conversation of the mechanism of action for day 2. Today we're going to talk about resistance. So as you can imagine, resistance is a huge problem. And currently, we have resistance to any antiviral. And pretty much any antiviral that we will probably discover, we will have resistance to it. Why? We have a lot of high mutation rates, right? If we think about the replication of viruses, we get lots of viral progeny. So their reproductive life cycles are a lot shorter than ours. And of course, some of the things that we are especially concerned with are going to be chronic infections. And anyone who is on extended therapy. And so that's going to be hepatitis B virus, HIV, of course, for hepatitis C virus. patients tend to be on extended therapy, although we can cure that, and we'll talk more about that in day 2. But we're most worried about these chronic infections, these persistent infections, oftentimes because the virus itself is at first integrated into the host genome. And so one of the things I think that is really important to remember is that viral resistance to every known antiviral has been detected. And that's definitely a very sombering statement. But again, resistance is a huge problem, and that kind of takes us to this idea of, well, which viruses do you think mutate and evolve the fastest, and why? And so I'm going to give you the answer first because we can't be here to discuss it, but let me know in the comments below if you are on board and if you are able to get to the same conclusion. So RNA viruses are definitely the ones that mutate and evolve the fastest, and in particular, single-stranded RNA. So double-stranded DNA is definitely much more stable. DNA itself is more stable, and a lot of it goes back to the sugars. So for deoxyribose and for ribose, that is going to be based on what is found at carbon 2 of the sugar. So if you have OH of hers, that is found in RNA. If you have an H, that is found in DNA, so in deoxyribose. And having that OH group on the second carbon of hers makes the ribose less stable. So in general, RNA viruses, RNA is less stable than DNA, and so they mutate and evolve the fastest. So we can actually talk a little bit more about these RNA viruses versus DNA viruses and kind of thinking about why. And of course, that first reason goes back to the idea that RNA is less stable, but also RNA viruses tend to have those error-prone RNA polymerases. And so when we say error-prone, we of course mean low fidelity, and they typically have no correction mechanism. So to kind of put into perspective what that means that you get one mistake every 10 to the 4th to 10 to the 5th nucleotides, and that is 10 to the 6 times more error-prone than DNA polymerases. And so what that translates to is if you have a genome that is about 10 kb, you're going to have one mutation every 1 to 10 genomes, which is pretty amazing. And this is part of the reason why we get so many mutations so quickly and RNA viruses can evolve so quickly. Now, keep in mind that these are random mutations that occur, and it takes one random mutation in just the right spot to get resistance. So something to keep in mind is that not all mutations are bad. So for example, for the first SARS epidemic that we had, there was actually a mutation that resulted in a deletion of part of an open reading frame. And what we found was this mutation actually decreased viral replication in some cells, and in general, it debilitated the virus. And part of the reason that virus was able to spread of HERS was it was still able to infect humans. It wasn't being outcompeted by anything else, but we actually think that part of the reason why the original SARS epidemic kind of died out a lot faster than this current one had to do with that deletion. So speaking of coronaviruses, even though we said that RNA polymerases don't have a correction mechanism, coronaviruses actually have special proteins. So they, of course, have the largest RNA genome, and these viruses have proteins that actually help with proofreading, and they help with keeping mutations lower. So even though RNA viruses are less stable because RNA is less stable and kind of more error prone due to their DNA polymerases, there's an exception to that no correction mechanism, and that, of course, is with the coronaviruses. So to wrap up for today, we're going to talk a little bit about DNA viruses. So when it comes to DNA viruses, and when we think about polymerases, of course, most DNA polymerases can excise and they can replace misincorporated nucleotides. And so because of this, they evolve less slowly. And in addition, we also see less diversity in DNA viruses. All right, so this is where we're going to stop for day 1. When we come back in day 2, we're going to pick up with part 3. So we've kind of talked about the idea of resistance, why it's important, how does it occur, and then when we come back in day 2, we're going to talk about mechanisms of antivirals, and we will do our best to also incorporate specific examples of how resistance occurs for the antivirals that we're talking about. So thanks for hanging out with me, and I will catch you in day 2. Bye, everyone.